Next one, cell division and colony formation. Cell division. So in the cell theory, I told you about the cell division. A cell division can be happen in two ways. Can you remember that one? Cell division can be happen in two ways. What are the methods that cell division can be happen? One is mitosis and the other one is meiosis. Mitosis and the meiosis. Okay, but in some microorganism, apart from this mitosis and meiosis, I told you they can reproduce by using a method called binary fission. Binary fission. Binary fission means that microorganism can produce another cell by using that method. Another cell by using that method, that binary fission method. Understood? Binary fission means producing another cell. That kind of thing. Clear?
Yes, sir.
Okay. Then what is mean by a colony? Colony. A colony. Colony mean actually here what happen is in our when we do some microbiology practicals, we make colonies. We make colonies. Colonies mean we find some bacteria or we find some microorganism and let it to grow. Let it to grow. It make several cells. It makes several cells and form a colony. Colony mean in that colony there are numerous of bacteria cells. So it can be a fungal cell or virus cell. So virus is there. Can you understand what is mean by colony? Of population of microorganisms. That means a population of a single microorganism is known as a colony. Understood?
Yes, sir.
When we doing our microbiology practicals in university, we used to make these colonies. We make colonies to identify the characteristic, actually the physical characteristic of these microorganisms. After we grow that microorganism in a specific slides, or this kind of watch glass, we can easily find the physical characteristic of that microorganisms. Physical characteristic of that microorganism. Okay? That is called the colony formation. Colony formation. And some bacteria have endospores. Endospores. Some bacteria have endospores. So these endospores are used to exist in unfavorable conditions. Understood? These endospores are used to exist in unfavorable condition. If there is any harsh condition is prevail for this that means for this bacteria, okay, for this bacteria, and to prevent from that hard condition, it makes a endospores. Endospores mean a thick wall-like structure. A thick wall-like structure. This a this a microscopic image of endospore. Microscopic image of an endospore. Clear? Do you want to explain it again?
Yes, sir.
Okay. So that means the environmental conditions are environmental condition different from time to time, no? Different from time to time. Sometimes have very higher atmospheric temperature. Okay, sometimes have a very cold condition, and when there are bacteria, this bacteria have to live in some acidic conditions. So those are known as harsh condition, no? Or those are known as unfavorable conditions. Understood? We also cannot live in that kind of condition, no? Or we also can't live, or we also harsh to live in that kind of environment. Understood? To spend that kind of harsh, or to spend that kind of very unfavorable or very harsh conditions, they make a thick wall behind this thick wall, not behind, thick wall outside of the cell wall. If the cell wall is this one, the cell wall is this, they make a thick wall like this. This is known as the endospore. So because of this wall, this bacteria can spend the unfavorable condition without any damage. Understood? Or still not?
No, understood, sir.
Okay. Let's write. Let's write. Right? Endospores are the endospores are the endospores are the special endospores are the special protective coat, special protective coat, special protective coat, special protective coat, dash, dash, special protective coat, dash, a thick cell wall, a thick, not cell wall, a wall, thick wall made by the bacteria, made by the bacteria, made by the bacteria, made by the bacteria to spend, made by the bacteria to spend, to spend unfavorable conditions, to spend unfavorable conditions, to spend unfavorable conditions. Okay. Actually, most of the students think the endospores are responsible for the reproduction. That is utterly wrong. Okay. There is no relationship between endospores and the reproduction. That is wrong concept. Okay. Next one. Bacterial impact on environment and other organisms. Bacterial impact. Can you tell me this is a advantage or disadvantage? That means advantage conditions or disadvantage conditions are caused by bacteria. That means other bacteria are advantageous or disadvantageous to us.
Both.
Both. Both. Both. We have benefit and disadvantage. That means advantage and disadvantage both from bacteria. So beneficial effect. First one is natural material recycling, primary producers, Industrial application, protein production, nitrogen fixation. Okay, then I want to give you an activity. So can you write one example for this each point, one example for each point. I will give you 2 minutes. Can you write? Because these are in our oral sources. Write as soon as possible. No need to write lengthy sentence, okay? Just one or two words are enough. Only for the beneficial.
Sir, do you mean I have to write examples?
Finish writing.
So primary products producers, what does it mean,
Primary
sir?
producers mean the organism which can do photosynthesis. That's a special kind of bacteria called cyanobacteria. And purple sulfur bacteria and green sulfur bacteria, those can do photosynthesis. We are talking about bacteria, okay? So we have to give some example by using bacteria. However, here the examples are given. No need to write. If you don't know about primary producers, just write here ability to photosynthesis. Ability to do photosynthesis. Okay, did you write something for the first time? Natural material recycling?
Yes, sir. Decomposing natural materials like dead
Yes.
bodies.
Decomposing natural material. materials. Okay. Then industrial application. So what are the industrial application you know? We have to
Yogurt
uh,
production,
yes, yogurt production. Yogurt production. Then alcohol production. That mean beer, wine.
product,
Sorry,
dairy milks production,
sorry, dairy. Ah, okay, okay. Dairy production. Dairy production. Okay. Then what else? That is enough because we have to learn. Then protein production. What are the things you know about protein production? Can you remember? I told you about a bacteria called E. coli. E. coli. Give me a second, okay? Okay. This is bacteria called E. coli. By using this E. coli bacteria and by using the method called genetic engineering, we can produce some proteins. We can produce some proteins. And do you know, have you heard about insulin? It's an enzyme, no? Insulin enzyme.
yes, sir.
So, what is the responsible or what is the function of insulin enzyme?
Production of insulin inside the body,
Yes, no, I'm asking the function of insulin.
controlling the sugar level,
Yes, controlling the sugar level. Controlling me, this can, we have some blood sugar level. No? If the blood sugar level exceed the normal level, the extra sugars are converted to glycogen and stored in our kidney and muscles. Kidney and muscles. Then after that, if it is that means if that amount is cannot be stored in kidney and muscles, that means if there are extra something after storing of kidney and muscles, it is converted to fat. It is converted to fat. Okay, next one is nitrogen fixation. Nitrogen fixation. What is mean by nitrogen fixation? I told you rhizobium bacteria can do nitrogen fixation. So what is mean by nitrogen fixation?
controlling nitrogen in soil,
Controlling nitrogen in soil. Uh, yes, it can
nitrogen,
control the nitrogen in soil. That is okay. Sorry?
nitrogen cycle,
Yes, it contribute to nitrogen cycle. That is also correct. But I want the exact definition for nitrogen fixation. Nitrogen fixation means what is the nitrogen percentage of air? What is the nitrogen percentage of air? Nitrogen percentage of air. It is around 70%. No? It is around 70%. So, then tell me do we use some input to provide nitrogen for plant? That means manure, something like that. Do we use some manure to introduce some nitrogen to plant?
Some fertilizers.
Yes, I'm asking about fertilizers. Yes. That means we use some fertilizers to give nitrogen for plants. No? So, do we provide carbon as fertilizers? Do we provide carbon as fertilizers?
No,
So, what is the reason? Why don't we provide carbon as fertilizer? The reason is we can, or the plant can absorb carbon as carbon dioxide from the atmosphere. So, why? So, in the atmosphere, there is only 0.03% carbon dioxide, no? Amount carbon dioxide, no? But in the atmosphere, the nitrogen percentage is around 70%. So, but we use fertilizers to provide nitrogen. So, what is the reason for here?
because the absorption of nitrogen in soil is higher
No, no,
by other plants.
that is not the reason. So the actual reason is, although there is higher amount of percentage in the atmosphere, I'm talking about nitrogen in that atmosphere, the plant cannot absorb that nitrogen. Plant cannot absorb the nitrogen because in nitrogen molecule, that means in nitrogen molecule means there are two nitrogen atoms. So it is bonded by triple bond. So this triple bond has actually to break this triple bond, higher amount of energy is need. Have you heard? Ever heard when lightning? Okay, when lightning happen, the soil gets some nitrogen. Have you heard that kind of sentence? When lightning, soil get nitrogen. Yes or no?
I haven't heard it.
Okay. Actually, there is some instances that lightning has very higher amount energy, that energy can break down this bond. That energy can break down this bond. Okay. So when it comes to the soil, it makes different kind of nitrate and nitrite ion. So that kind, so at that ionic nature, that means when nitrogen, that means when N2 is converted to this nitrate, NO3 2 minus or NO3 3 minus form, that means that form can absorb by the plant. That form can absorb by the plant. So nitrogen fixation means converting nitrogen gas into this nitrite or ammonium ion. Understood? Only these ions can be absorbed by the plant. Only this type of ions are absorbed by the plants. Understood?
So why don't plant absorb directly nitrogen from the soil?
Directly nitrogen from the soil. Reason is this one. When we take the nitrogen molecule, it is in this form, no? That means these two nitrogen atoms are bonded by uh, triple bond. To break this triple bond, high amount of energy is required. So plant cannot give that much of energy to break this triple bond. Understood? When lightning, what happened? These bonds are broken. These bonds are broken. So I told you there is some bacteria called rhizobium. This rhizobium have nitrogenase enzyme. Okay, nitrogenase enzyme. By using that nitrogenase enzyme, they can convert this atmospheric nitrogen into this kind of forms. Understood? That is known as the nitrogen fixation. Clear or not?
Clear, sir.
Okay. Then nitrogen fixation, some bacteria contribute to nitrogen utilization. Let's write some example, right? Rhizobium, RHI, BIUM, rhizobium. Then Acetobacter, Acetobacter, Rhizobium Acetobacter, TOR, sorry, TER, Acetobacter, Rhizobium and Acetobacter. Actually, actually, there are a lot of bacteria which can do the nitrogen fixation, but in our syllabus, only this two is given. Only this two is given. But in the all syllabus, there were some more bacteria. This is when you are doing some past papers, you will go through that one. For now, this is enough. Okay. Then so give an example, no? So let's write what is mean by nitrogen fixation in here. Let's write nitrogen fixation in here. Right? Nitrogen fixation is the process. Nitrogen fixation is the process. Nitrogen fixation is the of process of process of converting atmospheric nitrogen into converting atmospheric nitrogen into converting atmospheric nitrogen into converting atmospheric nitrogen into plant absorbable plant absorbable plant absorbable nitrogen ions. Plant absorbable nitrogen ion. Plant absorbable nitrogen ions. Finish?
Yes, sir. Done.
Okay, then let's write the simple equation. And to here, let's write nitrogenase. 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 And I will write NO3 minus, then NO2 minus, then NH4 plus, ammonium. Actually, this is not directly converted to this kind of form, okay? This understanding is enough. That's why I gave you this equation.
Okay, sir.
Okay, next we have to talk about harmful effect. First on it, food poisoning. Food poisoning example, Clostridium botulinum. Clostridium botulinum, this produce botulinum poison. botulinum poison. Next one is pathogenic diseases. So there are a lot of diseases caused by bacteria. No? Example, Mycobacteria tuberculosis. This tuberculosis disease is caused by this bacteria. Clostridium tetani. This causes tetanus. Then Vibrio cholera. This is Vibrio cholera. This is called cholera disease. Venia carotovera. Soft rot in carrots. Soft rots in carrot. As I can remember, I included. Oh no, no. I thought I have included this Venia carotovera disease. In our old syllabus, there were a lot of example for diseases caused by the bacteria. But in this one, only I have to give this for. Do you have any doubt?
Sir, should we remember this microorganism name? Like, is it a
Uh,
must?
Microorganism name, yeah. You ask me about the scientific name.
Yes, sir. Like the specific name
Mm.
which is belong to that
Yes,
division.
yes, yes. Yes, that is required. Actually, no need to remember the spelling in here also. In here, okay. I don't think spelling will be checked, and that means you are asking what is the microorganism. Sorry, what is the microorganism which cause tuberculosis? Write the scientific name. I don't think that kind of question will be asked for LOL SFT paper, but in some part, there are some microorganisms you have to remember the scientific name with the spelling. Okay, in here that is not necessary. That is not necessary. If you can remember, that is good. That's very good. Very good means that is excellent. And I don't think you have to remember this one. So in our next lesson, we have to talk about the industrial uses of microorganisms. No? So in that case, in that lesson, there are some microorganisms you have to remember the scientific name. Other than that, not enough. Not enough mean not need actually. Okay. Next one is cyanobacteria. No? Oh, we don't have enough time. Then shall I stop from here?
Okay, sir.
Mhm. I will need another 1.5 day. I think within 1.5 day I can do this one, do complete this tool. Then I will move to the 4.3 lesson, industrial use of microorganism. Then after completing that lesson, I will do physics part with 4.4 lesson. 4.4 lesson with physics. That means Newton laws and the force part. I'm going to start. Okay. Because we need to do that lesson mm, for 3 weeks. Let's see. Let's see. See, in our school, I had to take around 1.5 month to complete that first lesson. 1.5 month mean for 1 week, I have around 14 period. 14 into 6, 84 period. Let's see. However, we cannot take that much of time. Mm, I will tell you what we can do. I have actually I have planned to do some extra classes, but from tomorrow onwards I have seminars because from next year the syllabus is going to we changed, no? So there will be some seminars, so I have to attend them. That is compulsory. However, I have planned to do extra class on uh, Friday or Saturday this week. Let's see to do an extra class on the next week. Okay, and then shall I wind up the class? I will send you the paper, okay?
Okay, sir.
Okay,
Thank you,
then
sir.
okay, you're welcome. Good night.